Bismillahirrahmanirrahim. Assalamu alaikum, my dear friends. My name is Asen and I'm a YouTube lecturer of genetics. So today we will discuss about nano sequencing. So nano sequencing, first we must know about sequencing. What is sequencing? Sequencing is the alignment of nucleotide is known as sequencing. For example, A, T, C, G, G, C, T, T, A, A. This is the alignment of nucleotide. It's simply known as sequencing. So we come to history of sequencing. That what is the history of sequencing? So I little bit add some history of sequencing. And first, the first attempt and the history of sequencing is the Friedrich Master. In 1869, he isolated DNA in the form of chromatin. The second attempt in the history of DNA sequencing is Watson and Crick and Franklin. In 1953, he discovered the structure of DNA as double helix. In 1965, Robert Halley and colleague sequenced yeast RNA. In 1972, Walter Fair's sequenced first whole gene coding for MS2 virus protein. In 1975, Friedrich Sanger introduced plus and minus method for DNA sequencing using gel to separate newly synthesized DNA by size. So in 1977, Friedrich Sanger established the chain termination method and used it to sequence first complete genome of bacteriophage 174. In 1984, Fritz Foll developed non-radioactive sequencing platform. In 1987, Larry Hurt and ABI developed a semi-automated DNA sequencing machine in 1987. In 1995, first bacterial genome sequenced H. influenza. In 1996, Roneki, Yellen, and Nyreen introduced virus sequencing, a sequencing by synthesis method. ABI introduced first commercial sequencing using capillary electrophoresis. It was in 1996. In 1998, Sulixa developed sequencing by synthesis method that used fluorescent dye and we see a legal genome as sequencing in 1998. In 1999, first human chromosome number 22 as sequencing. In 2002, complete mouse genome is published. In 2005, the 454 system based on pyro sequencing became the first next generation sequencer to come on the market. That was the 454 system based on pyro sequencing in 2005. In 2007, Solid system which used sequencing by ligation is launched. In 2008, whole DNA sequence of a cancer is decoded for the first time in 2008. In 2011, Pacific Bioscience launched first commercial product, PacBio-RS, based on single molecule real-time technology. In 2012, and at the last, Oxford Nanopore technology commercialized nanopore sequencing. So it is little about the history of DNA sequencing. So the first attempt for the scientist to discover the cell, it was the first attempt, and the first difficulty for scientists is to discover the cell. And the second attempt and the second difficulty for the scientists is to find DNA. What is DNA? What is made from? What is it made? It was Watson and Crick that he discovered DNA. And the third difficulty, the third difficulty for the scientists is to find what is DNA made from? It's nucleotide, and the for difficulty for the scientists as to sequencing it, what DNA is actually made, and to find the nucleotides alignment, to find the nucleotides chain, that what is DNA made from and what is the arrangement of nucleotide. It was the last difficulty for the scientists to sequence the DNA. So today we sequence the code of life, DNA and RNA within second, because it is the struggles of all the scientists that today we able to sequence the code of life, DNA, RNA within seconds. And due to Oxford nanopore technology, so today we will sequence DNA RNA with a sequence, so it is in the form of nanopore sequencing. So what is nanopore sequencing? Nano means small and pore means house or for. And the sequencing of nucleic acid DNA RNA by passing the polynucleotide strand through small pore in the presence of ionic and electric current. It's simply about nanopore sequencing, that we sequence the nucleic acid DNA RNA by passing the polynucleotide strand through small pore in the presence of ionic and electric current. And these are some devices by molecular sequencing for expert nanopore technology. One is line gels, smedgine, menoine, gradine, and methine. The most common device for expert nanopore technology is menoine. Menoine is actually made of actually contain fluoxyl, sensory chip, and reagents. Fluoxyls is simply in the device menoine. There are some part which is known as fluoxyl, and this fluoxyl sensory chip is located, and this sensory chip area of microscope is located, mean nanopore is located here. Uh, this is simply about menoine, and which is connected to monitor any other devices. Uh, for us is and first we isolate DNA RNA, then isolated DNA RNA are transferred into menoine. And the most interesting thing is that it doesn't need PCR. It is the most interesting thing is that it doesn't need PCR. Here the DNA is formed from small nanopore in the presence of ionic and electric current. The most interesting thing in this that it is to feel ionic and electric current. Due to this field, we get some mm, we get some disturbance due to the nucleotide AATCCGG current is disturbed, mean that when we pass single strand of DNA from this nanopore, it contain electric and ionic current. When we pass the nucleotide chain ATTC, this 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 disturb the flow of current. And this disturbance is detected by the sensor chip and come with this message to the monitor, where it is shown in the form of AATCCG, where it is converted into groups, and each group represents its own nucleotide in the form of ATCGG. So this is simply about menoine, how the menoine process the DNA strand. So first the DNA strand is passed from the nanopore, and this contain two field. One is ionic and the rest is electric. By passing this DNA from this nanopore, the